Welcome, colleagues, researchers, clinicians, and knowledge seekers alike. This lecture is part of the TheraClaims Master Series, under the Unified Master Scientific Protocol. Presented by Bryan Geniuson, Research Systems Architect and Independent Translational Research Scientist, this series translates molecular mechanisms of natural bioactive compounds into structured, evidence-tiered health benefit frameworks grounded in systems biology, molecular pharmacology, and translational research. In this review of quercetin, we will examine the scientific evidence supporting claims related to antioxidant activity, inflammatory regulation, vascular support, blood pressure modulation, immune signaling, exercise recovery, metabolic balance, and emerging antiviral hypotheses. We will evaluate these claims across vascular, immune, metabolic, and redox regulatory domains, while clearly distinguishing between strong human evidence, moderate findings, mechanistic plausibility, and speculative extensions. Quercetin is a naturally occurring flavanol widely found in onions, apples, capers, berries, and leafy vegetables. Structurally, it contains multiple hydroxyl groups that confer redox modulating capability and interaction with inflammatory signaling cascades. Structured abstract. Quercetin influences inflammatory transcription factors, oxidative stress pathways, endothelial nitric oxide signaling, and mast-cell stabilization. Mechanistic and preclinical evidence is extensive. Human clinical evidence demonstrates modest effects in blood pressure modulation and selected inflammatory endpoints. Translational magnitude remains context-dependent. Disciplines involved include redox biology, vascular physiology, immunology, nutrigenomics, molecular pharmacology, and clinical translational science. Molecular mechanisms. Quercetin inhibits NF-kappa B activation and downregulates pro-inflammatory cytokines including tumor necrosis, factor alpha, and interleukin-6. It modulates MAP kinase signaling pathways and influences NRF2 activation, enhancing endogenous antioxidant enzyme systems such as superoxide dismutase and glutathione peroxidase. It also supports endothelial nitric oxide synthase activity, contributing to vascular relaxation mechanisms. In immune cells, quercetin demonstrates mast-cell stabilizing properties and modulation of histamine release. Systems integration. Within vascular tissue, quercetin may support endothelial function and modestly influence blood pressure. In immune signaling networks, it contributes to inflammatory tone regulation. In metabolic tissues, it interacts with oxidative stress pathways and inflammatory mediators. Exercise-related research suggests potential attenuation of exercise-induced oxidative stress markers. Clinical biomarkers. Human studies report small reductions in systolic blood pressure in some populations. Select trials show reductions in inflammatory markers, such as C-reactive protein. Exercise studies demonstrate modest improvements in oxidative stress proxies. However, effect sizes are generally moderate rather than dramatic. Claim landscape classification. Tier A strong. Human clinical support. There are currently no broad therapeutic claims for quercetin that meet strong, consistent human outcome validation across large populations. Tier B, moderate or mixed human evidence. Modest systolic blood pressure reduction. Exercise recovery support in certain contexts. Selective inflammatory marker modulation. Tier C, limited human evidence with strong mechanistic support. Endothelial nitric oxide pathway support. Mast-cell stabilization and seasonal immune modulation. Metabolic inflammatory signaling modulation. Redox and antioxidant pathway enhancement. Tier D, theoretical or emerging claims. Cancer prevention or treatment. Neurodegenerative disease prevention. Direct antiviral treatment efficacy in humans. Cardiovascular event reduction. Clinical versus mechanistic gap. Quercetin exhibits strong mechanistic plausibility across inflammatory and oxidative pathways. However, translation into large magnitude human clinical outcomes is limited. Cellular potency does not necessarily equate to systemic therapeutic impact in human populations. Bioavailability and translation constraints. Quercetin undergoes significant first-pass metabolism. Circulating forms are often metabolites rather than the parent compound. Absorption varies by formulation. including glycoside versus aglycone forms and specialized delivery systems. Plasma concentrations achieved in human studies are substantially lower than concentrations used in many in vitro experiments. Inter-individual variability, microbiome interactions, and dosing context influence outcomes. 4-dimension validation summary. Mechanistic certainty is high. Preclinical evidence is extensive. Clinical relevance is moderate and context-dependent. Translational readiness is moderate, constrained by bioavailability considerations. Key highlights. Quercetin is a mechanistically rich flavonoid with broad redox modulating capacity. Human effects are measurable but generally modest. Evidence supports cautious, support-oriented claim framing rather than therapeutic positioning. Proper interpretation requires distinction between cellular evidence and human outcome magnitude. Regulatory safe claim framing. Supports antioxidant defenses. Supports vascular health. Supports a healthy inflammatory response. Supports immune system function. Full scientific literature references and structured evidence summaries are available in the video description. A structured research companion document is available for members, including bioavailability comparisons, metabolite mapping, dose-response grids, claim classification matrices, and cross-system pathway integration analysis. In conclusion, quercetin represents a biologically active flavonol with strong mechanistic depth and moderate clinical effect magnitude. Within the TheraClaims framework, it exemplifies the importance of distinguishing mechanistic plausibility from clinically meaningful outcomes. Scientific, medical, and educational disclaimer. This content is provided for scientific, educational, and informational purposes only. It is not intended to diagnose, treat, cure, or prevent any disease. Statements regarding health effects are framed within structured evidence evaluation and may include mechanistic, preclinical, observational, or clinical research data. Interpretation should occur within appropriate clinical context and in consultation with qualified healthcare professionals.